Welcome to Lesson 5 in this series of hormonal communication presentations. This one is going to be on the structure and function of the pancreas. A quick look at our learning objectives shows us that we're going to need to discuss the histology of the pancreas, which basically means the structure of the cells within the pancreas, and we're going to consider how we might be asked to apply our understanding to a photograph of a section of pancreas to be able to draw and identify the structures there. So this would be linked, for example, with PAG 1, which involves microscope drawings. So if we start with the position of the pancreas within the body, it's found just behind the stomach in the upper abdomen. So just behind the stomach means that it's pretty central in the body. In terms of diagrams, it usually gets drawn as a kind of leaf-like structure, and it's fairly small compared to quite a lot of other parts of the body, but incredibly important. The pancreas has two very major important functions. The first one is as an endocrine gland. That's producing and releasing hormones into the blood, and that's what we're going to be focusing on quite a lot in the next lesson. And as an exocrine gland, producing and releasing enzymes which go through a duct into the duodenum. If we look at the pancreas in a little bit more detail, what we can see is that we have got a tube running down through the middle of the pancreas, which is called the pancreatic duct, and that at this end will join up with the bile duct, which is leading from the gallbladder and is carrying bile from the gallbladder, which has been made in the liver. And you can see, in fact, the pancreatic duct and the bile duct merge together just before it enters the duodenum, which is that sort of pink structure that you can see there on the left, and the duodenum is carrying food from the stomach. Within the pancreatic tissue, we can see a range of different types of cell. And what we've got here circled is we've got the acinar cells, which are the cells which are secreting enzymes into the pancreatic duct. And over here we've got this cluster of cells coloured in blue, which are the islets of Langerhans, which contain all the endocrine cells which are secreting the hormones. The pancreas has a role as an exocrine gland, which means it's really involved in digestion. So we'll just have a quick look at the digestive system, and what I've done is I've labelled up all the major bits of the digestive system, which are structures that I'm sure you're aware of, and I'm just going to now focus on any of the other parts of the digestive system which work as exocrine glands. We've got the liver, which produces quite a lot of chemicals, one of the main ones being bile, and the gallbladder, which stores that bile and carries it down through the bile duct. We've got the small intestine, which is divided up into two major parts, the duodenum and the ileum, both of which produce enzymes which are an important part of the digestive process. The salivary glands here on the right are producing salivary amylase, which is an enzyme which is breaking down starch. And then we've got the stomach, which plays a very important role in the breakdown of pepsin and also secreting various other hormones and other important things which are also linked in with it as an exocrine gland. And lastly, the pancreas, which is working as an endocrine and an exocrine gland, but in this instance we're talking about the secretion of pancreatic amylase and pancreatic lipase, trypsin, and a range of other enzymes which complete the digestive process. Summarising this information then, most of the pancreatic tissue is exocrine tissue, which means it's producing digestive enzymes. It's also producing this fluid called the pancreatic juice, which is very alkaline, and that's going to neutralise the gastric juice which has been created in the stomach, which is very acidic. We've got the clusters of acini, one acinus, many acini, and you can see that they are in little groups with a central gap in the middle, which is a duct, and these enzymes are going to be secreted into that duct, and then all these ducts will join together to form the pancreatic duct, which carries the pancreatic juice into the duodenum. This takes us on now to the pancreas as an endocrine gland. It has two very important hormones, insulin and glucagon. And what we're going to do is we're going to look at the histology, the cellular structure of the islets of Langerhans where these hormones are produced. This is an islet of Langerhans, and what we can see here is that we've got clusters of cells which are surrounded by a large number of capillaries. As the alpha and beta cells, which are within the islets of Langerhans, are secreting their hormones directly into the bloodstream, you need a large number of capillaries to be efficient and to carry it away exactly as it's needed. So as I mentioned, we've got these two types of cell. We've got the alpha cells, and the alpha cells here are releasing a hormone called glucagon, and the A in glucagon indicates that it's from the alpha cell. The beta cell, on the other hand, is producing insulin, and I'm afraid I've got no way of helping you remember that in particular, other than it's obviously not an alpha cell producing glucagon. We're going to have a quick look at this diagram of an islet of Langerhans and just the labels here. So at the very top we've got pancreatic acini. So these are our clusters of cells with a gap in the middle, which is going to join together to form the pancreatic duct. We've got the alpha cell here, which is in sort of dark purple stained, and the beta cell, which is in a sort of light pink stain. And we've got another labelled cell here called a delta cell, and we don't need to know anything about the delta cells for the this particular course, but it's just to be aware that this is a cell which is producing various other chemicals which are moderating the activity of the pancreas. And lastly, we've just got this label here, which is just identifying the whole group as the islet of Langerhans. Histology is what we see when we look down a microscope and we can see the cells, and being able to identify and label those cells is important. So what we need to be able to do is we need to be able to look at a photograph of pancreatic tissue and identify the main structures that you can see. So we're going to just do that here on this diagram and this photograph, so we're going to compare the two. So to start with, hopefully you can recognise we've got this lighter region here, which is the islet of Langerhans. Now generally speaking, they are very noticeable because they tend to be a little bit paler in colour, and they are dotted in amongst all the pancreatic tissue. Over here we've got the acini, which are secreting the enzymes. So these tend to be darker. You can just about see, if you look carefully, that there's a sort of like a space in the middle of them, sort of in the centre of each of these little clusters, and these ones are going to be producing the enzymes. And just over here we've got a capillary, and the capillary is noticeable because you can see we've got some red blood cells inside there, and I think you can even see a white blood cell, but that is what helps you to identify that it's a capillary, and you'd expect to see a lot of those sort of dotted in amongst all of this tissue. We're just going to compare the islets of Langerhans and the pancreatic acini. From an appearance point of view, the islets tend to be lightly stained, whereas the pancreatic acini tend to be darker. The islets tend to be large and spherical, certainly quite rounded and quite noticeable as a cluster, whereas the acini tend to be very small berry-like clusters, but they make up the large majority. The islets of Langerhans are producing endocrine material, so this is the endocrine part of the pancreas, whereas the pancreatic acini are the exocrine part of the pancreas. The function of the islets of Langerhans is to produce and secrete hormones, and the function of the pancreatic acini is to produce and secrete digestive enzymes. Looking at this photograph here, we can see that the purple stained cells are the beta cells, and we can see some lighter sort of pink stained cells in amongst those, and those are the alpha cells. What I would just say as a word of caution here, we cannot assume that if they're purple or pink that they are one cell or another. You would have to be given information about that in order to be able to identify that on a diagram. And here's just another example, but you can see in this example, although the islets of Langerhans are noticeable simply because of the fact that it's sort of in the centre there, and it's like a slightly lighter cluster in the middle, it isn't so easy to see because the staining hasn't identified them quite so clearly. One of the things that you should be able to do at some point is some practical work where we give you a slide and we ask you to do a plan of the slide to identify the islets of Langerhans and the acini and so on. And then we might even ask you to do a high-powered drawing of an islet of Langerhans. And so if we were doing this in a lesson, we'd be giving you a sheet to complete, and there'd be some instructions on there about how to go through this whole process and to tell you how many labels to put on. But your normal drawings of microscopes would apply here. Hopefully at some point you will get a chance to do this. We're going to finish off this presentation with a few questions. So to start with, what is an endocrine gland? Give 2 types of stimuli that trigger hormone secretion. Once the hormone is in the bloodstream, why doesn't it affect every cell in the body? And thirdly, a question about adrenaline, which we'll come back to in a second. So what I want you to do is have a look at those questions, just pause this presentation for a moment and just note down your answers, and then move on to have a look at the answers which I'm going to cover now. So question 1, what is an endocrine gland? A group of cells which are specialised to secrete hormones. Question 2, 2 types of stimuli which trigger hormone secretion. You should hopefully have got the change of concentration of a specific substance, for example like change in glucose, or another hormone, or possibly electrical impulses. And once a hormone is in the bloodstream, why doesn't it affect every cell in the body? This is because the hormone can only bind to specific receptors because it has the complementary shape for that receptor, and it cannot bind to anything else. Question 4. When adrenaline binds to receptors in the cell membrane of liver cells, it activates the enzyme adenylyl cyclase. This then catalyzes the production of a second messenger inside the cell. What is the name of the second messenger produced? What effect does the second messenger have inside the cell? And what is the first messenger in this example? So the answer for this is the second messenger is cyclic AMP. The second messenger activates a cascade of enzyme reactions to make more glucose available to the cell by catalyzing the breakdown of glycogen into glucose. So adrenaline is effectively increasing our glucose concentration in the blood. And what is the first messenger in this example? That's going to be adrenaline. This question is an application question. I'm not going to read it through, but I'm suggesting you have a quick look at it and have a go at the questions that follow. And when you're ready, what I'd like you to do is I'd like you to just go on with the rest of the presentation and find out if you've got the answers right. Now this brings me to the end of this particular presentation, and the next presentation is now going to be looking at the way in which the hormones released from the pancreas, insulin and glucagon, actually control the blood sugar. So we're going to be looking at the way in which they work upon the body to control the levels of glucose in the blood. Thank you for listening, everybody.